In just about every medical drama, you probably have seen a machine which displays this particular waveform on its screen. This is an EKG or an electrocardiogram. This is a visual representation of the electrical activity that's occurring in the heart. In this particular video, we're going to talk about the heart's electrical system and how an EKG is used to measure that electrical activity and what we can learn from the results. The heart's pumping action, the cardiac cycle, is regulated by an electrical conduction system. This system coordinates the contraction of the various chambers of the heart. The action of the heart works a little bit differently than other muscles. Skeletal muscles, the muscles that are connected to your bones, are special in that their operation is voluntary. Basically, our brains have to be able to tell those skeletal muscles to respond. They're going to be stimulated by receiving a signal from our nerves. Whereas the cardiac muscle is self-excitatory and involuntary. There are specialized cells within the heart which are actually able to generate the electrical signal, which allows for the coordinated movement of the heart. And that happens without us even thinking. So electricity is everywhere, even in our human body. Now, our cells are actually specialized to conduct electrical currents. The electricity is required for things like our nervous system to send signals to the body and the brain, and for our heart to be able to pump. So the elements in our bodies, like sodium and potassium and calcium, they have a very specific electrical charge. Now, almost all of our cells can use these charged elements called ions to generate electricity. Now, the contents of the cell are protected from the outside environment by the cell membrane. The cell membrane, as you remember, is made up of phospholipids that create that barrier so that only certain substances can cross to reach that cell's interior. So not only does the cell membrane function as a barrier to molecules, it also acts as a way for the cell to be able to generate that electrical current. At rest, cells are going to be negatively charged on the inside, while the outside of the cell is mostly positive. This is due to a slight imbalance between the positive and negative ions inside and outside the cell. Now, the cells can achieve this charge separation by allowing for the ions to flow in and out of the membrane through special proteins. Now, it is the flow of those ions across the membrane which generates that electric current. So cells will control the flow of specific charged elements depending upon what proteins are present on the cell's surface. When a cell is stimulated, it allows positive charges to enter the cell through open ion channels. The inside of the cell then becomes more positively charged, which triggers further electrical currents that could turn into electrical pulses, called action potentials. Our bodies will then use certain patterns of action potentials to initiate correct movements, thoughts, and behaviors. For example, an action potential generated by the specialized cells of our heart is what's going to allow for that signal to cross the heart and initiate a heartbeat. The actual electrical stimulus for our heart is generated by specialized tissue, which is found up in the right atrium of our heart, and it's called the SA or sinoatrial node. This sinus node will generate an electrical stimulus about 60 to 100 times per minute under normal conditions. Now, the electrical stimulus generated by the SA node is going to be transferred to special cells that are in the atria, which are able to conduct that electrical impulse. The whole atria is going to get bathed in that electrical signal, and once it's fully engulfed the atria is going to contract. Now, we want the atria to contract separately from the ventricle. So that signal is going to be slightly delayed before it starts to move into the ventricle to allow for the atria to contract separately from the ventricle. The signal is going to be picked up at the base of the atria in another group of tissues, which is called the atrioventricular node, or the AV node. The signal then is going to be transferred to conductive tissues, which are in the ventricle, the bundle branches, and then the Purkinje fibers. Once that signal has passed through the bundle branches and the Purkinje fibers, then the ventricle is going to contract.
Action potentials originate in the sinoatrial node and travel across the wall of the atrium from the sinoatrial node to the atrioventricular node. Action potentials pass slowly through the atrioventricular node to give the atria time to contract. They then pass rapidly along the atrioventricular bundle, which extends from the atrioventricular node through the fibrous skeleton into the interventricular septum. The atrioventricular bundle divides into right and left bundle branches, and action potentials descend rapidly to the apex of each ventricle along the bundle branches. Action potentials are carried by the Purkinje fibers from the bundle branches to the ventricular walls. The rapid conduction from the atrioventricular bundle to the ends of the Purkinje fibers allows the ventricular muscle cells to contract in unison, providing a strong contraction.
In order to measure the electrical activity that is happening in the heart, an EKG can be done. When we examine the electrocardiograms from a healthy individual, we're going to see a series of telltale waveforms that is referred to as sinus rhythm. Each element of the waveform has been identified with a letter identifier. So we have our P wave, a QRS complex, and a T wave. The first waveform, the P wave, is caused by the electrical activity that is occurring in the atria as it prepares to contract. This is the electrical activity of depolarization as the ions are moving into the cell to prepare for contraction. The QRS complex is a much larger waveform, and this is the electrical activity that's happening in the ventricles as it's preparing to contract, as those ions are moving across the membrane, further depolarizing those cells in the ventricle. Another telltale waveform of our sinus rhythm is the T wave. The T wave is the electrical activity that's happening in the ventricles as it's returning to its resting state. This is when the cells of the ventricles are repolarizing, as we're returning to the point where the outside of the cells are going to be positive and the inside of the cell is going to be more negatively charged. So if the waveforms are showing us the electrical activity in the heart as it's preparing to contract, when do they actually contract? Well, the atria is going to contract in between the P and the R waves, which we refer to as the PR interval. And the ventricle will contract in between the S wave and the T wave, something we call the ST segment. As I mentioned before, a normal heart rate is called a sinus rhythm. The sinus rhythm is maintained by the actions of the SA node. For this reason, the SA node is oftentimes referred to as the pacemaker of the heart. Using an electrocardiogram, we can observe various different rhythm types. If a rhythm is detected that doesn't follow the typical waveforms of PQRST that are identifiable, we say that this is an arrhythmia. It's out of rhythm. It's abnormal. When you put a or an in front of a word, we're saying that it's not, so it's not in rhythm. Something is going on. Sometimes the heart can have a normal rhythm, but its speed can be different for one reason or another. Sometimes you can have an abnormally slow heart rate. For example, if somebody is in a state of unconsciousness or that is very, very cold, then they're going to have a heart rate perhaps of less than 60 beats per minute. And this is referred to as bradycardia. If you exercise, your heart rate's going to increase, and the heart will be experiencing a sinus tachycardia when it's beating above 100 beats per minute. If an arrhythmia is detected, sometimes the arrhythmia is actually just self-correct, while others may require medication to maintain an appropriate rhythm. In other cases, external defibrillation is going to be necessary. This can be done in a hospital setting, or if constant monitoring needs to be done, it's possible to implant a pacemaker, which will put electrical connections near the nodes of the heart to be able to monitor and correct small arrhythmias. Isn't it amazing that our cardiac muscle cells have the capacity to generate and conduct electrical signals on their own, which allows them to do their job every day?